Recording.
Good.
Bienvenue à tout le monde au séminaire d'aujourd'hui. So welcome everyone to today's QCAM seminar. We have the pleasure to host Laura Fabris. So Laura is an expert in nanomaterial synthesis and applications, and she will discuss that in greater details today. Briefly about her background. So she has done a bachelor's and a master's and doctorate of chemistry at the University of Padova in northern Italy. She then moved to the United States for a postdoctoral stay at the University of California, Santa Barbara, and was hired right after that at the University, Rutgers University in New Jersey. So she has developed a very vibrant group that combines nanomaterial synthesis, characterization techniques, and applications, and mostly uses metallic nanoparticles in her research. So she will discuss today about nanomaterials chemistry approaches to address health and energy challenges. So as Matteo and Peter said, so if you have questions, please ask them in the chat or at the end by raising your hand at the end of the talk. So thank you very much, Laura, to take time to discuss of your research to us, and we're looking forward to your talk.
Thank you, Jean-François. Thank you, everybody. Good morning. Bonjour. I don't speak French, unfortunately, so this is as much as I can say to you today. But thank you for being here today and for hosting me to this for this seminar today. And I'm looking forward to talking to you about our research and to having you ask me any questions and any curiosity you might have on our work. So before I move on, because I tend to always rush towards the end, so I want to acknowledge the people that have done the work. And in this very 2020 group photo here, you can see some of the people that have really been the greatest contributors to this work. Primarily, this is my postdoc, Dr. Hao Wang, who just left my group about a week ago to move to Duke University in North Carolina. But a lot of the work is being also carried out by other students who graduated with me and then left. Dr. Supriya Atta, who's now also a postdoc at Duke, Ted Sulas, who is a postdoc at EPFL, and Manjari Bamidipati, she left academia. She's now in a science communication company. And as I said to you, how worked with me for over 3 years and did a lot of great work with me. And the majority of the work you'll see today has been sponsored by DARPA Biotechnology's Office, US National Science Foundation, and several Bush Biomedical grants that Rutgers University provided me over the years. So our group, you can see some of the details of our research on our website. Here it is wrote up in the right corner. So our group is sort of divided into two main areas, if we want to say, those who enjoy mostly the fundamental science would are focused on colloidal synthesis, of which you see a lot of examples today, plasmonic nanomaterials, electron microscopy characterization. As you can see here, this is a 3D tomographic reconstruction of one nanoparticle. So this is not a cartoon, this is how the particle looks like. And we have also been working on finite element modeling to really understand how the plasmonic response, the optical properties of the nanoparticles correlate with their morphology, and how we can use them sort of in a feedback loop to improve the synthesis of the nanomaterial, drive our design for any specific applications we are interested in. And from the standpoint of the applications, we have focused for the longest time. So if you had known me for a while, you would know that, probably my group has been working primarily on surface enhanced Raman spectroscopy, in particular for diagnostic with respect to some diseases and for sensing, the development of sensing platforms. But recently, we've gotten very interested in photocatalysis, especially hot electron-driven photocatalysis. And I'll tell you a lot of the details of this work today. So, all right. So as the title says, what we do here is really driven by the realization that there are many problems affecting both the health and the energy, frameworks in our daily lives. not only in our daily lives, but also there is a big divide between developed countries and poorer countries. If we look at the energy area, we realize that while the developed countries are really focused on developing clean, renewable energy sources to offset really pollution and the climate change, there are a lot of countries, especially in sub-Saharan Africa, they are completely off the grid. And so if you see into this map, you realize that 60% of the people in 47 countries in sub-Saharan Africa are completely off the grid. They have no access to electricity. And so we might think, that it's inconvenient not to have light at night, but this has a lot of repercussions also in the safety of these people and their health. So a few years ago, I had the opportunity to sit in at the United Nations in New York City for a meeting on women health, women's safety. And what was brought to my attention is that in certain countries where there's no light, there's no electricity, it becomes very dangerous for women to really care for their families, provide water, provide food, because they can get attacked along the way, it gets dark early. So this opened up my eyes into the need not only to address the needs of the Western countries, but maybe primarily to really contribute to the well-being of people that are much less fortunate than we are. Along the same lines, you can look at a different side of the coin, and we realize that the growing population numbers bring a strain of resources from the health standpoint. So we obviously are now in a pandemic, and the pandemic, this virus is affecting our lives, so everybody's lives. But what we realize that, until now, what communicable diseases such as, let's say, influenza or other enteroviruses have been primarily affecting certain countries in the world. Again, sub-Saharan Africa, Southeast Asia, and these countries are really suffering for these reasons. And if you actually go and look at how people die in different parts of the world, people die differently depending on where they live. If we look at high-income countries, we realize that people die due to non-communicable diseases such as stroke, heart attack, Alzheimer's diseases. Whereas in low-income countries, of course, there are still the contributions of some stroke or other non-communicable diseases, but we realize that the majority of them are communicable diseases. We have diarrheal diseases, which is a communicable disease, HIV/AIDS, tuberculosis, malaria. So how do we provide a contribution to solve both richer countries' issues and low-income countries' issues? So we have to design approaches that address both communicable and non-communicable diseases. Clearly, nobody can solve all issues, but as a nanomaterials chemist and a spectroscopist, I can really think about ways in which I can contribute, my group can contribute to addressing these issues. And so today we will see how we can look into these different directions. On the one hand, we can look at how we can use gold nanoparticles to address and improve photocatalyst performance. And I'll show you how we can produce solar fuels such as hydrogen in situ under solar light illumination. And on the other hand, we can use nanoparticles to improve the diagnosis of both communicable diseases, such as in this case, influenza A, and non-communicable diseases as prostate cancer. Over the years, we've worked on many different plasmonic nanoparticles, but recently we have settled a little on these type of nanoparticles that we define nanostars. However, if you look at nanostars and you do a literature search, you're going to see many different shapes that are not exactly identical to one another, not similar. The protocols are very different from one another. So today I'll show you two specific categories of nanostars, both seed-mediated, created through seed-mediated synthesis. One type is like this one with multiple sharp spikes. And the other more recent type we're working on is less spike, like maximum 7 spikes, but very elongated, which allows us to tune their plasmonic response well into the near infrared. So if we think about nanostars, if we think about plasmonic nanoparticles with respect to the problems we're trying to address, we can see two different really areas of interest. On one hand, if we look at hot electrons and hot electron photocatalysis, we realize that upon absorption of radiation, from biplasmonic nanoparticles, there is the generation of hot electrons, a high number, or relatively high number of hot electrons that have large energies. So these hot electrons, highly energetic electrons, if they can be extracted from the nanoparticles, then they can do things. For instance, reduce molecules and do chemistry, reduction chemistry in the surrounding environment. And on the other hand, because we are interested in surface enhanced Raman scattering, these nanostars can be really relevant because upon absorption of light, they can scatter the electric field, and this electric field can be substantially enhanced. And as you can see here, this image is a 2D slice of a 3-dimensional finite element model that we ran on these nanoparticles that has 5 spikes in plane and 2 perpendicular spikes coming in and out of the plane of the screen. And so what you can see here, and then we'll go into more detail, but what I want to point your attention on is that these red areas here, which represent the highest region of field enhancement, are very broad, right? Extend very far in the surrounding environment, which is really interesting. We have done some work where we've seen that these extension can be up to 13, 15 nanometer out, which is much larger than what we see in spherical particles. And also that we have some interesting behavior inside the particle here. I'm not going to talk about it today, but we hope that we can find collaborators in the future that can help us really study what happens at the single particle inside these nanostars. However, what we have learned through the study of these nanostars is that we need to really focus on the synthesis of nanomaterials. A lot of nanostars have been designed and been applied, but if you look at really fundamental work on these nanoparticles, you'll be surprised that is the fundamental science on the synthesis and really the plasmonic characterization is not as widespread. So over the years, my group has been particularly interested in colloidal synthesis of plasmonic nanomaterials, really going into the details of understanding how these particles grow. And so in various different projects, we have looked at the synthesis of gold nanorods. And in particular, in this particular paper, we identified what is the growth mechanism and what is the evolution of their crystallographic structure over time that leads to thermodynamic stabilization of the particles. But we have also focused a lot on synthesizing nanoparticle assemblies, in particular nanoparticle dimers, both of spherical nanoparticles or of nanorods, because what we know is that the junction between these nanoparticles is where the highest field enhancement can occur. This is called hotspot, the region around or nearby a nanoparticle where high field enhancements can be achieved. And so as I was saying right now, we know that when we have spherical nanoparticles, if we bring the 2 nanoparticles together, up to 1 nanometer gap size, we see substantial enhancement at the gap. We cannot go below 1 nanometer because then we start having electron tunneling effects. But at these levels of separation, we have high enhancement, which then tapers down at larger separations. However, if you do try to synthesize these particles, then you realize that it's not as straightforward. You have to have the absolute control of the kinetics of the reaction, the thermodynamic the reaction. You have to control surface charge of the nanoparticles, concentrations of the linkers, temperature, concentration of the nanoparticles present. So it takes some time to really optimize it. And we did a lot of molecular dynamics simulation studies to really understand the conditions. On the other hand, well, we found out that if we make nanostars, they are going to carry their own hotspots. So this is relatively more convenient because we synthesize the nanoparticles and we don't need to assemble them with anything else. And so learning from this, we have decided to really embark in a work where we not only decide to synthesize gold nanostars, but we really try to optimize their monodispersity. the idea about pushing monodispersity so much is the fact that whether we do ensemble measurement or single particle measurements, whether we look at cell environment or we look at a catalyst, if we ensure that the nanoparticles are all exactly morphologically the same, we know that their behavior, if we ignore the concept of polarization, light, the angle of incidence of light with the nanoparticle, if we exclude that, we know that their response is going to be uniform. And then if we see any inhomogeneities or heterogeneity of the data, we know that that's due to the system we're studying and not the nanoparticle that we're using to enhance the performance of the technique we're using. So we started looking at the synthesis, the seed-mediated synthesis of gold nanostars. This seed-mediated synthesis uses employs a spherical seed and uses ascorbic acid as the reducing agent, obviously silver nitrate to impart anisotropy to the spikes, but also Triton X100 as a surfactant. The use of this surfactant is very interesting because it really allows us to really reduce drastically the number of the spikes with respect to what I showed you before, leading to a nanoparticle shape that has maximum 7 spikes. And I'll tell you why maximum 7 spikes. Not only that, what we saw is that if we increase the amount of silver nitrate, not only we can really tune the length of the spikes, but we can maintain monodispersity. So now it's kind of difficult to see for you in these slides, but looking at this diagrams, these bar diagrams, we can see that the distributions are very narrow with standard deviations of about 2-3% or even lower in some instances. And so this is very interesting because yes, these particles are not yet perfect, but achieving this monodispersity is very difficult when you have high shape anisotropy and these very, very anisotropic shape, right? So there is one question though that we keep asking ourselves: what is monodispersity in this case? We know that if we have a sphere, as long as the particles have the same diameter, that's monodispersity. Nanorods, we look at aspect ratio, but what do we do here in these nanostars? So we can look at dimensional parameters of the spikes like we did here, but also we need to start thinking about the number of the spikes, right? Their orientation. Are there any defects? Why some spikes grow and some spikes do not grow? So going forward, we are very interested in understanding this aspect as well to really optimize and possibly be able to scale up the synthesis of these particles so that they can become relevant. One of the most interesting things that we gather from this work is to understand, or at least have a beginning to understand the growth mechanism of these nanoparticles. So what we realized in this study is that starting from the spherical seed, that is a single crystalline seed, the key step that needs to occur to produce this nanostar is the evolution of the seed from a spherical single crystalline seed to a 5-fold twin seed that has a dodecahedral shape. So what do we see here is that we have this dodecahedral shape seed, 5-fold twins here, and the presence of silver nitrate and Triton X100 is necessary for the particular growth because these two entities will absorb selectively on selective positions or selective facets on the growing seed, leaving only the corners here, the 7 corners available for growth. So that's where the 7 spikes come from. Obviously, as I said, there's going to be defects and this is something we have to deal with, but ideally that's what we should expect. Another interesting thing is that if we look at the growth of the spikes, right, we see that these 5-fold twinning or the morphology of these particular corners from which the spikes grow is retained. And as you can see, these are the heads or 2 tips that we've analyzed. And another interesting thing that we have observed, if you look at this right side of the slide, is that if we have low amount of silver nitrate, so therefore low amount of metallic silver on the nanoparticle, we will see that if we start at short times after synthesis with this 5-fold twin, If we don't have enough silver, we have the reshaping completely of the spike, and also the loss of the fivefold twins. And so you can see here this is one spike examined at different times. And as we can see here, that there's a completely reshape and restructuring of the spike. So the interesting thing that we gather from this study is that not only silver is necessary to impart the anisotropy to start growing the spikes, but also is necessary to keep them from reshaping, to keep them from shrinking over time. And this is very important for these type of nanostars, because obviously the small ranges of curvature means that these positions on the nanoparticles are very highly energetic. And so we don't want them to really give up to thermodynamics and reshape very rapidly. What if we did then some STEM EDX analysis, and what we observed is that we can identify where the silver nitrate, or actually the silver, metallic silver, goes on the stabilized nanoparticles. And you see here at very low concentrations of silver nitrate, it basically deposits primarily, and you will see a bare monolayer on the surface of the spikes. But if you start increasing the amount of silver, we see that this monolayer becomes more and more uniform. And then if we go to the maximum amounts of silver nitrate, we see that this silver migrates down into the crevices of among the tips, and that's where it's going to settle here in this position. So there is a lot more to understand, but this is very interesting because it does indeed confirm that silver is not an irrelevant component of this nanoparticle, and confirm some of our previous results that we have collected with ICP studies, where we had identified that there's a huge concentration of silver in these particles, and we didn't know why. So now we finally know where it ends up going. And a very interesting aspect of these nanoparticles that we first were able to model, as you can see here, this was a model that we actually developed without knowing a priori if we were ever be able to develop this nanoparticle. And what we had found in this model is that there are 3 resonant bands that are associated to the spikes that we can observe. We call them first, second, and third harmonic. You can call them, multipolar modes. We decided to call them this way because we found a resemblance in antenna theory, in the separation of the resonances depending on the morphology of the antenna, on the properties of the antenna. But what is interesting is that this first harmonic mode is very intense, and actually it explains the highly intense field localization that we saw before in that 2D slides I showed you before. And very interesting for us is that these resonances really move very far into the shortwave infrared regime, which could be relevant in the future, not only for applications in biology, but also for antennas for devices or filters or these type of applications. What is also interesting is that if we collect spectrophotometric data of these particles and we look at the experimental ensemble experiments, what we see there is a relatively nice match between the position of the spikes. There can be something to be optimized. We can narrow the width of the spikes, but from our standpoint, we believe this is a nice result that we were able to achieve. We did one extra step. We decided to go and look at the single particle from the single particle standpoint. We decided to go and look for these modes, for the first, second, and third harmonic modes. And we did that using electron energy loss spectroscopy. We know very well that we cannot exactly compare photon-based excitation with electron-based excitation. There is a momentum, sort of differences here. We need to take this into account, but semi-quantitatively, we can find that there is a nice match between the results. And that was really exciting for us. For instance, what we did, we took this nanostar and we probed 2 spikes into 2 different modalities. We probed the aloof mode, where the electron beam is shown far from the spike. When we probe the intersecting modality, with actually the electron beam really crosses the spikes. And so what we saw is that in the ALUV modalities, which is a 5 and 6, we clearly can probe the first harmonic mode, and in one case, the second harmonic mode. And this is also the case for the intersect approach. However, we see some slight deviation in the resonant peaks. What is interesting is that in these two positions, in position 7 and in position 5, so position 7 and position 5, we see only the first harmonic mode. And what is interesting about that, because our model had predicted, hey, in position 5, that the majority overwhelming resonant mode would be the first harmonic. You remember the red halos there. And in position 7, we would have a node in the resonant mode. And so we should not expect to see the second resonance in this position. So going forward, we would like to really extend these studies not only to the nanoparticles bare, but also perhaps coated with other materials, coated with metal oxides, coated with polymers, and see how these interactions at the interface play a role in this particular studies. So learning from these fundamentals properties of these nanostars, and sort of based on some of nice work that Sasha Govorov and collaborators did back a couple of years ago, we started really thinking about how can we look at these nanoparticles, not only the 7-branch nanostars, but also the multi-spike nanostars, for applications where we can leverage hot electrons, because there has been evidence that, theoretical proposed, that the hotspots or the spiky positions in these nanostars are positions where large numbers of hot electrons are generated due to breaking of momentum conservation. And so we said, what if we actually can extract these hot electrons from those spikes? And sort of, what can we do with them? So, there was, and I have to admit that this project is the one that actually is the most exciting for me to have brought to completion, because basically what happened is my graduate student and I really sat down brainstorming on this type of catalysis work, and we really wrote down on paper what we expected was going to happen, and exactly everything that we wrote, really we measured and we gathered from our results. So it was very exciting from that standpoint, because it doesn't happen very often that this occurs. We always have to really rethink many things oftentimes. So what we decided to do, we decided to really look at photocatalysis. And with my collaborator, Dr. Fuad Celik, he's an expert in photocatalysis using titanium dioxide. So titanium dioxide is very useful for water splitting, for instance, for many other reactions, but for instance, in water splitting. And so if you look at photocatalysis for water splitting, what people normally do, they create a bulk, photocatalyst, TiO2, and then they decorate it with nanoparticles. But what we decided to do is to really flip the coin. And we still could create this metal semiconductor interface, but in a little bit different way. So the metal semiconductor interface is important because if you have a plasmonic nanoparticle, the nanoparticle absorbs radiation. Some hot electrons are excited about the Fermi level, some of which have high energy. If they have enough energy and the Schottky barrier, the interface is designed so that they can be transferred outside the nanoparticle into the semiconductor, then we can put them in the conduction band where they actually can do things, all right? And so this was our guiding principle. We have to generate hot electrons. We need to extract them and allow them to do things. And the other way, as I was telling you, the other way to make them do things, to make nanoparticles do things, normally, the primary way to make nanoparticles do things is to excite in the UV, where you have a microscale TiO2 particles, microparticles or bulk TiO2 decorated with small size gold nanoparticles. These are about few nanometers. We are not interested in plasmonic behavior in these nanoparticles, but rather into their surface defects. So in this case, the TiO2 absorbs UV radiation. Then these electrons, the conduction band electrons, are transferred into the small nanoparticles, and that's where the reaction occurs at the surface. What we actually are more interested in, and we are more interested into the particle absorbing radiation through plasmonic resonances And then once, as I said before, the electrons are higher up in energy, they can be extracted and transferred into the conduction band of the photocatalyst. Normally, however, what is been done so far is that this excitation in first case looks at UV radiation. So this small sliver here, 5% of the solar spectrum. And if we look at the other spherical nanoparticles, we see that it focuses around 520 nanometers. So the visible, high energy visible range. But what about all the rest of the solar spectrum? We are actually wasting it. And so we decided to find a way in which, by leveraging the gold nanostars, we can absorb all the radiation into the broad spectrum, well into the near infrared or infrared, and do the same thing. So to do that, we had to really optimize materials design rules. And we did it this way. We decided, well, we thought the first thought we made was, well, if we have a macroscopic or microscopic TiO2 catalyst and we decorate it with nanoparticles, in the ideal case that this nanoparticle is spherical, we would have only one point of contact, right? From which the electrons can be extracted into TiO2 from the nanoparticle. We know it's not the case because the nanoparticles are multifaceted, but yet the points of contact would be minimal. So our idea was that, how about we take the nanostars and we actually coat them with TiO2? Can we do that? In that case, we have perfect contact everywhere, and any electron that is generated, we can take it out. However, to do that, we have to really follow these design rules. We need to obviously create the hot spots, create the hot electrons. We need to tune the resonances to be into the near infrared. We have to optimize the metal semiconductor interface to reduce at minimum any defects, to remove any capping agent, because that would really hinder the transfer of the electron. We have to create a semiconductor that is crystalline, but is also very thin, so that any hot electron that is extracted into the TiO2 can be, again, further extracted into the surrounding environment where it can run reactions, in our case, in the surrounding water. So if you ever tried to do this reaction where you can try to coat gold nanostars with crystalline TiO2, you know that you cannot do it the right way, the normal way. If you try to use salt gel protocols to coat nanostars with TiO2, then you're going to get amorphous TiO2. We need to recrystallize it with calcination at either 200 to 500 degrees C. But if you do that, you will get crystalline TiO2, but you will reshape completely the nanostar. These nanostars starts to reshape above 85 degrees C. So there you go, our limit is 85 degrees C. So we had to figure out a way to reduce the synthesis to below 85 degrees C. And so in this work, you can see all the details of all the synthetic protocols, but basically my student was able to develop a protocol that runs at 70 degrees C. And 70 degrees C and is able to create a TiO2 shell that is a rutile anatase mix, and as you can see in this XRD, and is actually very thin. And as you can see here is, you can say almost epitaxially growing. You can see, we're not exactly entirely sure, but we can see that the growth of TiO2 on gold resembles epitaxial growth. And you see this very smooth transition from the gold crystal to the TiO2 crystal from this interface. So these are, in my opinion, very beautiful images because it's very complicated to grow these nanoparticles. So now we have these nanoparticles designed, as we said, the gold nanostars at the hotspots, the spikes. We can generate hot electrons there. We have a crystalline TiO2 shell that is grows unhindered on the nanoparticle, and it's thin. So if we are able to absorb radiation, we should be able to extract the hot electrons into the surrounding environment. So we decided to run these reactions. We first ran some controls. We tried different illumination conditions. We started in dark, obviously, no illumination, UV, visible, and broad illumination. Broad illumination means anything from the UV on. It's just that we didn't have a particular filter well into the infrared. So we illuminated all, including UV, visible, and near infrared radiation. So obviously, if you have no catalyst, there's no production of, there's no reaction. If these two catalysts are non-optimized catalysts of the same morphology that I was telling you about, P25 is a commercially available photocatalyst. Anatase is only obviously crystalline TiO2, and this is only gold nanostars. As what you can see here, the best performance you can get is from P25, which is a commercially available one, and from these non-optimized photocatalysts that we designed. And as you can see here, we are about 350, 375 micromoles per grams of catalyst that have been produced under the different illumination conditions. So now what we do, we tried our nanoparticles, and what you can see, look at this yellow bar. We increased by almost 10 times the production of hydrogen in the same conditions by using our optimized nanoparticle. So this is really exciting because we saw that by really tuning in very high detail the properties of the nanostars, we can really produce hydrogen in water in situ. So imagine having this scaled up into production and delivered wherever obviously small energy consumption needs are necessary, but that could be very impactful in countries where there's no energy, no access to electricity. So now I'm really switching gears a little bit, and I'm going to tell you about how we can address health challenges using surface-enhanced Raman spectroscopy. And the first part I'll tell you about is how we can detect and monitor the progression of cancer. We use the concept of SIRS tags. So SIRS tags, how do they work? They have composed of nanoparticles, in this case gold, which are decorated by Raman reported molecules, Raman active molecules, which are covalently bound to the metal surface. Then you can code the nanoparticle with the reporters, either with silica, or if you don't want to use silica, just with a polyethylene glycol. And the important part is that the outermost terminus of the polyethylene glycol needs to be functionalized to carry targeting moiety. This is an antibody that can target a protein or a proteic biomarker, but you can have aptamers, which in our group we prefer. Aptamers are oligonucleotides that are based off RNA or DNA that fold in specific hairpin conformations and are designed to very selectively and strongly bind to their specific targets. And in some cases, they behave even better than antibodies. So in this case, what you do, if you have, let's say, a solution, or you have a tissue, or you have a cell where you are expecting to find your biomarker, you expose this biomarker to your SIRS tags, to these nanoparticles. You let them bind, and then you go and map the system to look for the SIRS signal of these molecules. And the SIRS signal would be a proxy for the identification of the particular biomarker. So these can be used in various ways. Some people, they use them in circulation, although, we know very well that the delivery of nanoparticles in vivo is not very efficient. We get up to 99.5% particles that are excreted by the body, and so it's not very efficient. What you can do is use them through endoscopy. You can, let's say you want to have a colonoscopy done, and you can inject the particles first as a contrast agents. They will bind to any, let's say, tumor that is present on the tissues of the colon. And then you use an endoscope with Raman capabilities to really look for the SIRS signal of these nanoparticles. They have been used also for monitoring tumor margins during open surgery, which is, in my opinion, this was a very impressive achievement that was published back in 2014. And also, in several ways, they are used to monitor biomarkers for disease. These biomarkers can be proteins, they can be DNA or RNA, they can be exosomes, they can be circulating tumor cells, and so on. In particular, we are interested in proteic biomarkers of disease and in genetic biomarkers of disease. And so our work focuses primarily on this last section. So why are we interested in prostate cancer? Prostate cancer is a very, we believe that, we have clinicians have a good handle on prostate cancer. However, what we realize is that prostate cancer is often overtreated. People are treated even if they don't need it. And one of the reasons why this happens is because prostate cancer tissues are very heterogeneous. And when you have histopathology alone to be used to assess the severity of the disease, this is very hard to accomplish safely. For instance, you see these 5 tissue biopsies. This is not our work, was taken from this manuscript, this paper here. You can see these represent 5 different patients at the same stage of the disease. And as you can see, these are very heterogeneous. So discussing with oncologists, it has become clear to us that they need quantitative metrics to assess the disease projection for these patients and to stratify them in particular classes of severity. So we use our nanostars as SIR-stacks for this purpose. So we looked at one specific biomarker, which is called PSMA, prostate-specific membrane antigen. We prefer this to PSA because PSA give contrasting results and can be expressed at high values even in patients for which the prostate was removed. So, is not very reliable, but PSMA has been hypothesized to be a very reliable biomarker. So we decorate our nanoparticles with Raman reporters, and we have PSMA aptamer to capture the PSMA protein wherever it is present. So we have tried 3 different approaches. We have looked at PSMA soluble protein. We looked at PSMA that decorates the membrane of cancer cells. And we have also looked at PSMA that is present on prostate tissue microarrays. So in prostate tissue microarrays, the biopsy tissues of various patients are collected, fixed and deposited on these glass slides. And you can actually quantify the expression of PSMA and use these numbers to stratify patients. And so by doing that, first we needed to determine that the aptamer, the PSMA aptamer works. And not only we found out that it works, but if we look at this particular middle section where we have functionalized both the aptamer and the antibody with FITC, and we monitor the presence of FITC fluorescence on these cells, we see that in the comparing by PSMA antibody stained cells and PSMA aptamer stained cells, we see that for the same cell population, we see a substantial more fluorescent signal from PSMA aptamer. So we not only find that it works, but it does work very well. And so we were assured that this was doable. And so we started, first of all, to monitor the expression of PSMA biomarker onto cells. And we use 2 cell lines. We have the LNCIP cell line, which is known to express high values, high numbers of PSMA, and compared to the PC3 cell line, which is shows to not express much PSMA. And this is known from the literature, but we obviously had to verify it. We verified it with using RT-PCR and Western blotting. And we saw here that exactly is what we found, that in LNCIP, we have expression of PSMA in both, using both studies, both tests, whereas in PC3, we have basically no expression detectable. And as you can see here, what we see here is that the signal here that we observe is the signal, the SIR spectrum of the Raman reporter that with which we had functionalized the nanostars. And so we looked at the intensity and the presence and the intensity of these peaks to really assess the presence of the PSMA and quantify it. First of all, we realized that by looking at different cell populations of the same cell type. So for instance, this is LNCIP population, different cell groups, and we average the signal from the different groups, we see that every group has different expressions of PSMA, which is in line with the heterogeneity of the disease. What we also found is that we could determine that the density of these biomarkers on cells, on individual cells. And we found out that LNCIP cells express about 45 PSMA molecules per square micron compared to PC3, which is less than 5 molecules per square micron. So this is very interesting because now if we're able to have different cells at different stages of disease, we can actually quantify the progression of the disease by looking at just one cell or like a small population of cells by looking at the density of these PSMA molecules. But the most important thing we did was to monitor patients. And so we first did the control study where we looked at immunofluorescence staining, because this is one of the standard protocols to use. We took the patients divided in 3 groups. Group 1, here you see the group 1 is patients that are being told to do wishful waiting. Wishful waiting is you do nothing and you hope for the best, which is, you know, not very promising, but these are supposed to be a low risk patients. Group 2 are patients that are medium risk, but they are told to do radiotherapy or start chemotherapy. Group 3 are patients at high risk, high severity of the disease. These patients have undergone often surgical removal of the prostate, or they have also initiated androgen deprivation therapy, which is a very, very drastic therapy and really affects the livelihood of these patients. So what we saw here by staining really these tissues and monitoring the fluorescence signal, we saw that one, the data points are very narrow, very clustered, so it's very difficult to discriminate the various patients. And also very interestingly, we see that this approach does not distinguish group 2 and group 3, right? So this is very, very serious because these patients can be at any stage of the disease and we cannot really separate them. But when we use SIRS, what we were able to observe is that there's some outliers. So this is important, and I'll tell you about why later. But also, if we don't consider this outlier, in the group 1, we see that the group 1 patients are all clustered at low level of PSMA expression. Then we have a slightly higher level. We have the group 2 patients, and group 3 is those that express higher levels of PSMA. The outliers, though, are very important because, for instance, these patients should not have been told to wishfully wait. It should have been told to really do something. So unfortunately, we don't know what the fate of these patients was. They were not consented patients, so they didn't disclose we didn't have access to their final outcomes, but this is concerning. So we are not saying that SIRS should replace the other techniques, but could be worked to really as a companion diagnostic technique to assess these patients. And so now I'm going to really go for the last few minutes I have, I'm going to really move into the last part of how we can use nanomaterial chemistry to address diseases and to address in particular communicable diseases. This is very relevant to our current situation because also the coronavirus is an RNA virus. We have been working on some diagnostic tools right now. We're very close to finishing up, but the data are not really clean yet, so I'm not going to show them. But we have moved a little into the direction of the coronavirus. And in general, keep in mind that this approach can be applied to any RNA virus going forward, for which you know the sequence. So influenza virus is a very interesting virus. I am no virologist, but have had the luck to collaborate with 4 very talented collaborators who are experts in virology. One of which in particular in influenza A, his name is Chris Brook, he is at the University of Illinois, Urbana-Champaign. And so what I've learned working with them is that influenza A is an RNA virus, as a virus that is divided into 8 segments. Each segment needs to be present to make a pathogenic variant. So for the virus to work, all 8 segments have to be present. Each of these segments codes for a specific protein in the virus. And so, for instance, we were particularly interested in segment 4. Segment 4 is the hemagglutinin HA segment. Hemagglutinin is the protein that is responsible for binding to our lung lining, the cells that line our lungs, through sialic acid. So it is the one that really attaches the virus to our lungs. It's similar to the spike protein in the coronavirus currently. What is also interesting about these RNA viruses, coronavirus included, is that through replications, they mutate substantially. In some cases, they give rise to very, very short sequences, very different sequences compared to their progenitors. And this causes genetic drift. Genetic drift is basically a deviation of the virus from its original genome, which and genetic drift leads the virus to evade the immune response. And most importantly, it was causes zoonotic spillover, which means that if an animal is sick and comes with this mutated, right, with drifted virus, and it comes into contact with a person, the person can acquire the disease. This is what is happened with coronavirus. So studying viral evolution and studying what happens is very important. So what we were working on in this project was to design nanomaterials, nanoparticle-based tools for intracellular identification of the presence of viruses and the presence of super spreaders. And super spreaders are those cells or individuals that are, for some reasons that we're not sure a priori, they are able to produce huge numbers of viruses during replications. So if we were able to identify with over a super spreader is, we would be able to isolate it to understand why the phenotype of the super spreader leads them to produce these viruses. So this is the really broad target of this project. But what I'll show you today is the development of these nanoparticles to identify the viruses intracellularly. So the segment 4, a hemagglutinin segment, is composed of about 2,000 nucleotides, of which the blue bars here represents the conserved ones, which means that over replications, they never mutate, they never change. The internal parts are always amenable to mutation. So what we told ourselves, well, if we target these conserved regions, we will always be able to pinpoint and quantify a virus wherever it is. And so what we did was to design gold nanostars. We partially coated them with silica because we saw that it can enhance the signal while providing stability. We functionalized them with PEG for biocompatibility, and also what we functionalized them with this oligonucleotide with DNA that is fluorescently labeled at the 3', thiolated at the 5', and also designed to have self-complementary flank regions at the termini, so that when the oligonucleotide is left alone, it folds into a beacon loop. Okay, it folds into a beacon loop, and in this position, the fluorophore is near the metallic surface. And because every time a fluorophore is near a plasmonic metal surface, it gives surge signal, whereas the fluorescence is quenched. So in this case, this nanoparticle produces a highly intense surge signal. Then what we did, we added a complementary oligonucleotide. We designed it in red, and this is made to complement, to hybridize with the blue beacon and stretch it out, so that the fluorophore is far, the surge signal is quenched, and the fluorescent signal is on. Finally, we take this construct and we expose it to the virus. And when this is exposed to the virus, the red oligonucleotide is designed to dehybridize from the beacon. The beacon is alone again, it folds back again, the fluorophore comes close to the surface, and then we see the surge signal all over again. So we go from on, off, on. And basically, we go from an off situation to an on situation whenever the virus is present in the system we're studying. We study buffer first. We see here when the beacon alone is folded, the surge signal is on. We hybridize it sorry, this is actually mislabeled. We hybridize it in black here with the beacon, right? And it quenches, so when it opens up. And eventually we expose it to the RNA target in red, and I'm sorry for having confused you, in red, and we see a recovery of the signal. So what you can see is that we go from on to off to on again, as I was telling you before. The recovery of the signal is not entirely 100% perfect because we believe there's some steric hindrance issues here at play that we're currently investigating. What is also very interesting is that if we introduce on purpose mutations in the target, for instance, well, here, this is the target as it is in the virus. Here we introduce 2, 4, 6, 8 mismatches out of about 24 nucleotides, so about 30% of mismatches. We can see that we can not only identify them, but that the particles are still robust and they're able to identify the virus even if it has mutated, which is really, really relevant for the purpose of diagnosing the virus. The last step was to bring the virus into a cell and see if it actually recognizes a virus present in the cytosol. So what we did, we took HeLa cells. HeLa cells are easier to transfect with plasmids. So we took three plasmids, one coding for hemagglutinin and two others coding for PB1 and PB2, which are two other segments in influenza A, for which though our particles were not designed. And so we saw what? When exposed to either non-transfected cells or to cells that were coded with PB1 and PB2, we see no signal. And when we expose the particles to cells that have been transfected with the hemagglutinin segment, the signal goes off. The red curve represents the case where the nanoparticles were coated with this polyelectrolyte, and this polyelectrolyte is responsible for producing endosomal escape. It bursts the endosome and releases the endosomes into the side and releases the nanoparticles into the cytosol. So we see a slight increase in the signal here. But this is extremely interesting because the cells are alive. We have loaded them with viruses and we can probe them with the nanoparticles. So going forward, we can think of really not only developing devices that probe cells, but also because these are very small concentrations, we can think that we are very hopeful that we can get really a diagnostic assay coming out soon for low viral loads with diagnostic relevance. So to conclude, I want to show you just that this technique can be adapted to fluorescence modality. The reason why we're trying to adapt it to fluorescence modality is because if you really want to enter the clinic or the biology lab, you need to do it very slowly. Using fluorescence is a way to transition because SIRS is, I know that for a fact from personal experience that SIRS is not well known in those environments. So by looking at fluorescence first, we will be able to really hopefully transition SIRS as well. And so these beacons can be designed to go from fluorescence off to fluorescence on, as we were discussing here before. And so you can see that the kinetics, the turn on kinetics of the fluorescent signal is very rapid at every concentration. And then we have a calibration curve here that is linear at low concentration, and which is very positive from the diagnostic standpoint at very low viral copy numbers. Finally, this can be again adapted to use fluorescent flares. And fluorescent flares, if you're not familiar with them, are nanoparticles that are functionalized with double-stranded DNA, for which the one that is thiolated and bound to the nanoparticle is complementary to the target, and the other one, which comes off eventually, is fluorescently labeled. So we go to a situation where the fluorescent flares are turned on, and in the presence of the target, they will turn off here. They will turn on in the presence of the target. And as you can see here, even though the turn on of the signal is not as sharp as before, still is able to identify the presence of the virus. So now I'm gone to the end of my talk, and so I hope that you found something. What I told you will be something you can find relevant for your interest or for your research. And my hope is that by focusing on nanomaterials chemistry, by looking at the synthesis with a little bit more detail than we have tended to do so far as a community, we will be able to really tune our applications much better, and hopefully to really bring solutions to these global needs. Thank you.
Thank you very much, Laura, for a very, very interesting talk, covering very interesting topics as well. So we can open the floor for questions. We already have one from Michel Grandbois. Michel, do you want to ask it live, or you want me to read it? I guess I'll read it on this. So relating to the discussion, detection of prostate cancer cell markers, could it be that the multifunctional aspects of the stars going on the particles could lead to more efficient molecular recognition at the cell surface, and thus provide enhanced contrast compared to fluorescent markers?
You mean the features that can expose the aptamer in the sense? That's if I understand correctly, that's the question.
if I understand correctly as well.
Yeah, it could be. And actually, this is very interesting. It's a very interesting question, because what we are trying to do now, we are trying by looking at selective functionalization of the nanostars, we want to really study how we can only functionalize them at the tips. So and then study how an aptamer at the tip, which is less hindered into its folding and unfolding, can interact with the biomarkers expressed on the surface. We can study clustering of these biomarkers. We know that there's no even distribution of biomarkers on a cell. They tend to cluster. And so this is a very good point. And we are very interested in really figuring out with high resolution what's happening there. That could well be one of the reasons.
We'll go for one raised hand. So Fiorenzo, if you want to turn on your microphone, and if you want also your camera, if you wish. And microphone still off.
Unmute. Okay.
There we go.
Fiorenzo, how are you?
Wow. Good. How are you?
Very good. Thank you for the excellent talk as usual. I have a question. I may have already asked you this at some point previously, but can you selectively attach other types of nanomaterials at the tips, sorry, the apex?
Yes, we do. We have done it before. We've done it with spherical nanoparticles.
Yeah.
It is a little bit of a nightmare if you don't coat the particle, right? And we tried in the past. We had a naked nanostar, and we tried to bind the smaller spherical nanoparticle with EDC NHS coupling. That works, and we can be region specific, but it's complicated, right? My students said, I told my student, just do it. It's easy. After a year, she comes back very angry because it took her a year to really, because you have to change buffers and etc. But if you have the silica coating, like we were able, so now what we do, we coat the particles with silica completely, and then we etch it. And we etch it only to the point where only the tips come out. So at that point, if you have a thiolytic chemistry, that will go only to the spikes, and then you can eventually etch completely the silica, because we etch it with sodium borohydride, so it's very mild. And so what you can do is you eventually end up with the decorated tips. Yeah.
Okay, that's fantastic. I'll send you an email.
Okay.
Thank you.
So we have another question from the chat. So is it possible that the precipitated silver on the gold surface works as seeds, so that nucleation happened there leading to the branches?
Well, it could be that it helps, right? That it could help the metallic silver at the corners of the seed, could help nucleating there. It could be something interesting to verify, but I am not entirely sure at this time. But yeah, I mean, we have proposed this mechanism, but we are not entirely sure, obviously, if this is the case. What would be really interesting, and what we hope we can get to do in the near future, is to try to look at more like liquid TM, to look at in situ growth to real-time growth, because this is a relatively slow reaction compared to other nanostars reactions, so we can actually see when things happen and see where they start happening. So hopefully we'll be able to get some more information.
Yeah, maybe related to that same topic. So I had a question of mine. So do you have to keep your nanostars stored in some sort of silver nitrate dope buffer to maintain the shape, or do they maintain the shape over a long period of time? Because you mentioned some reshaping that can occur, but I wasn't sure.
That's a very good point. I have not thought about it. That would be interesting to try it out. Normally we do not keep a high content of the silver nitrate, but that would be really interesting to see. That would be really interesting. So what we try to do is, this reshaping is really a big issue. So we commonly synthesize the particles close enough to when we want to move on with the next step. Usually, if you have polymer PEG or silica or TiO2, whatever it is, obviously that locks the shape. But this is really interesting. I'm going to really try to look into that. I'm going to set up a study, because it could be that this exchange, because silver oxidizes so easily, it could be this exchange that keeps them. Very nice point. Thank you.
There's still a little bit of time for questions from the chat or by raising your hands. But in the meantime, actually, if there's other questions, I have another one, if I may. So you showed that the nanostars coupled with the TiO2 actually raise the hydrogen evolution reaction to a fair extent. Do you know if it's because you you harvest more of the NIR from the plasma? And do you have any like spectral correlation to the improvement of the reaction, or is it just because of the electron transfer is more efficient? So is there a spectroscopy?
I think it's both. I think it's both. I think it's both, because when you look at the selective excitation at the different, we don't have single particle studies, which will be interesting to do. Sure. But yes, there is both. It could be an effect of both, but we tried to normalize, but being an ensemble, it was becoming a little bit, more challenging. But, we have, unfortunately, the student that did this work graduated. And so, in the lab, nobody wanted to do this synthesis. They're like, no, this is not going to work. So for the last, 6 to 8 months, we have not looked into that, but we are really trying to set up some nice experiments to study more in depth the mechanism. We're interested, for instance, in using the new nanostars, where you can really tune the resonance, the first harmonic band, very much. And then we can probably selectively excite at each of these resonances that are well separated and really study the dependence a little bit better.
Okay. Thank you very much, Laura, for these very, very good answers, and also for a very, very interesting talk. So I don't see any further questions from the chat or from the participants. So, well, the last thing to do is really to thank you very much for taking time to entertain us about your research, and I'm sure we'll spark discussions among the QCAM members. So thank you again, and
thank you.
We're looking forward to have you in person at some point.
Thank you. Yeah.